Last class, you have learnt properties of genetic material and semi-conservative method of DNA replication. This lesson is new for you because you haven't learned any of the basics of this lesson in your lower classes. So, I am planning to take each topic for one class. This will help you to understand the concept better. In this class, we will see only DNA replication machinery and enzymes. Let's see DNA replication machinery and enzymes. This process of replication needs a set of enzymes. Here, enzymes are termed as catalysts. DNA replication takes place in the S phase of cell cycle. Already you have learnt in 11th itself. This phase occurs during interphase before mitosis or meiosis and is responsible for the synthesis of DNA. Usually, cells will take between 5 and 6 hours to complete S phase. Let's see enzymes involved in this process. The main enzyme is DNA dependent DNA polymerase. It uses DNA template to catalyze the polymerization of deoxynucleotides. Here, template means a single strand of DNA that is used by the DNA polymerase enzyme as a basis of copying the DNA. Then next, polymerization. Already you have used this word in chemistry. Polymerization means several monomers combine to form a polymer or polymeric compound. Average rate of polymerization by these enzymes is approximately 2000 base per second. These polymerases has to catalyze the reaction with high degree of accuracy. Any mistake happened during replication which results into mutations. DNA polymerization is an energy demanding process. Because of that, deoxyribonucleoside triphosphate perform dual purposes. One is act as a substrate and another one is provide energy for polymerization reaction. Additional enzymes also required for this process along with DNA dependent DNA polymerase. They are helicase, primase, topoisomerase, SSB, ligase, etc. Let's see the functions of these enzymes one by one. Enzyme helicase. This enzyme unwinds DNA from tightly double-stranded structure only after the strands are separated. DNA polymerase can do its job of creating the new strands. This enzyme separates the strands by breaking the hydrogen bonds between the bases of two strands. Next is topoisomerase enzyme. This enzyme removes tension from uncoiled DNA strand. SSB. Protein stabilize uncoiled DNA strand so that it cannot coil again. The uncoiling of DNA strands form a Y-shaped structure known as replication fork. Next is primase. This enzyme creates a short fragment of DNA, that is primer, paired with the template strand. This enzyme initiates the process of creation of new strands. DNA polymerase cannot initiate the process. Because of that, it needs a primer to start the process of DNA replication. DNA dependent DNA polymerase catalyzes the polymerization only in one direction, that is 5' to 3'. It creates additional complications at the replicating fork. As a result, on one strand, that is template 3' to 5', the replication is continuous. This is known as leading strand, while on the other strand, template 5' to 3', it is discontinuous strand. This strand is known as lagging strand. The discontinuously synthesized fragments called Okazaki fragments. Later, these are joined by DNA ligase enzyme. Practice the given diagram. Here, primer acts as a starter of the process of replication. Then ligase helps to join the synthesized fragments.